Hello, everyone, and welcome to the Natural History Museum. For those of you who don't know me, I'm Sandy Knapp, and I'm a botanist here at the museum, and I've been here for longer than I can remember. I'm like that guy on Pirates of the Caribbean who's become part of the ship, which is a bit scary. And the reason that John and Imran asked me to talk today is because this Darwin's birthday party event started with Jim Mallett from UCL and I deciding that it would be a really good idea for us to get together, all us evolutionary biologists across London, for us to get together and to have talks that presented different points of view on the same scientific subject. Because sometimes in science, we get very channelized and we think about that there's only one way to think about something. And there's always more than one way to think about something. There's always more than two ways to think about something. And so Darwin's birthday party started out as a kind of let's have a party, but it also started out as a way of introducing everybody across the spectrum who was interested in evolution across London to thinking about things in a different way, or to think about things in a different way than they normally did. So the reason I'm up here blurbling about this now is because this is the 30th anniversary of the very first Darwin's birthday debate. And that was in 1994. And for an event which was set up to have people disagree, it was actually a completely damp squib. I mean, the title of it was Evolution in the Cambrian, Biology's Big Bang, or Just a Damp Squib. And actually what happened is the speakers, Derek Briggs and Simon Conroy Morris, agreed with each other that Stephen Jay Gould misinterpreted them completely. So they were in complete agreement. And through the years, there have been huge numbers of different kinds of speakers. We've talked about the Cambrian explosion a couple of times. We've talked about phylogenetic fuses. We've talked about conservation. We've talked about Haldane's law, which actually, interestingly, was the only one that actually happened on Darwin's birthday. Otherwise, we miss it one way or the other. We've talked about conservation genetics, evolution and development, species. But one of the things that's important about this year as well is this is also the 20th anniversary of the first time that Nick Barton was invited to speak at Darwin's birthday party. He's the only scientist who's been invited twice. So I think that makes this a red letter day. And in looking at the kind of details of who spoke and what the topics were, it struck me that one of the really gratifying and exciting things that's happened over these 30 years is that the proportion of women who speak at this event has gone up. Overall, it's only 21%, but in the last 10 years, it's closer to 50-50. And I think that's really a good thing for our field. And it's really a good thing for society in general, that we're really thinking about diversity in a good way. So just to say that one of the things that is so wonderful about Darwin's birthday party, I think, is that this was an idea that Jim and I had, and we thought, oh, this might be a good idea, this would be fun, but mostly it was an idea about having parties. But actually, it's turned out to be something that's bigger than that, because it's brought all of you people here year after year after year to listen to talks which are a little bit outside your field, perhaps are things that you don't think about every day. And that's really was the core of the idea behind it. And so it's really gratifying that it's kept going for 30 years. And so may it keep going for another 30 years in what may one day actually be a slightly better Flett auditorium. So I want to thank you all very, very much. And thanks to Jim Mallett as well, because without him, this would have never happened. Thank you.
So thanks very much, Sandy, and welcome everyone. It's my job as one of the co-directors of the Centre for Ecology and Evolution to welcome everyone here, and also to thank our very generous sponsors for making these events possible. We now have rolling funding from the Royal Society, who see our organisation as a fantastic way, as it indeed is, for London-based researchers to come together, discuss critical ideas in ecology and evolution, and to form collaborations and friendships as a result of that. So thanks very much to the Royal Society. I think some of them are definitely going to be a table. Is anybody in the room from the Royal Society? Well, we have a fellow of the Royal Society here, of course. But the science communication team of the Royal Society will be outside. You have a chance to chat with them and talk about all the great ways the Royal Society funds science, particularly early career researchers, which is what a lot of the CEE is based around helping. We've also this year had fantastic funding from the Genetics Society, who see this as another way of catalysing these interactions between young researchers. And that's meant that we've been able to invite or pay for the expenses of people to come from outside London to come to this event. Hands up if you've come to London to come here from outside London as a recipient of Genetic Society funding. So there should be about 8 or 9 of you. Congratulations, you're here because of the Genetic Society, so thanks very much for that. They also gave us funding for helping people with caring responsibilities to attend, as well as funding for early career researchers to come along to our dinner, which is later on this evening after the wine reception. So that's fantastic. Thanks very much to the Genetic Society. The Natural History Museum are hosting us very generously. So thanks to Sandy for introducing us, and to Imran and Esther for helping to organise the whole event, along with Amy Godfrey, who provides a lot of admin support from UCL. We really wanted the 30th anniversary event to really be a chance for reflection of 30 years since we started these, or Sandy started these events, 20 years since Nick last spoke, so he's got a great perspective to present us about this. I first came to a CEE event from Leeds in 1995, and it was John Maynard Smith and Stuart Kaufman discussing adaptive landscapes. And I remember going home fully astonished that I'd managed to be in the same room as these amazing people talking about ideas with such grace and candor and kindness. And I think that's a very, very important aspect to the way that we discuss science. So we thought today what would be a really nice thing is to kind of come at the discussion about genetics and genetic and phenotypic evolution from these two kind of opposing ends, if you like. One end being how alleles spread in populations, how they get established in populations and spread through populations across species ranges to form the kind of communities that we so value, that are part of the biodiversity that is responsible for the economies on the planet. And also from the other end of the distribution, think about the discipline, if you like, of behavioral ecology, of understanding how those genotypes make phenotypes that affect their fitness throughout different life stages and across different environments, different sexes and different tissue and cell types. So we're really hoping that we can get this reflection of the way that these fields have changed and how they might change in the future. And I hope if any of us are still doing science in 30 years, if science is still being done in our societies in 30 years, let's hope, we will be able to look back and think about what we learned over the past 30 years and reflect on 30 years of a lot of fun discussion, if not necessarily hugely tangible progress all of the time. But that's what science is all about. So thanks again very much for coming. Do please become members of CEE if you're not. We're very much looking to include more students and postdocs and early career researchers in our committees. If you look at this QR code, you'll take us to the CEE website, find out more about the 4 events that we organise each year, this being the Darwin's birthday one. So thanks very much, everyone, for coming. I'm now going to ask Richard Nichols to introduce our first speaker today, Nick Barton, who will be followed by Ashley Griffin. So, thank you, Richard.
Okay, what can I tell you about Nick that you don't already know? He's obviously a very impressive scientist. I'm not going to reel off his list of prizes and awards because it would be too long, and my introduction will go on for too long. I'll concentrate on one. He was made a Fellow of the Royal Society at a very early age, 39, I think, 38 or 39. And I heard a story about that, which I'll tell you, which was that before the Royal Society noticed that their fellows had a disproportionate number of people with Y chromosomes, they noticed they had a disproportionate number of old people. And they decided they were going to remedy this by finding the best early to mid-career scientists in the country. So they asked the academies, academicians from around the world to recommend British scientists. And they were astounded with the unanimity of which they were given one name. And it was Nick. Something else you might not know is that in addition to his remarkable work on evolutionary genetics, mathematical genetics, he's a prodigious in the field. He's worked on grasshoppers, he's worked on snapdragons. If you've ever been out in the field with him, you'll realize that he brings his mathematical understanding to field work, in particular that the shortest distance between 2 points is a straight line, whether or not it has brambles up to your waist. Final thing I say about him is that despite the fact that he works in remarkably difficult areas, applies very innovative mathematics, He's exceptional at explaining that to a more general audience. So if you haven't read his review papers, get back and do that. I think you'll see that in the talk which you're just about to hear.
Hey, thanks, Richard, for the slightly embarrassing introduction. And also to John, I'm not sure whether I interpreted the past and future of genetic and phenotypic evolution correctly, but I think there may be many interpretations, and I'm not sure I'm going to talk about what you think I'm going to talk about. I wonder how I'll get to my presentation. There it is. It's amazing. The evolution of evolutionary biology. That doesn't give you much of a clue either, does it? So just to sort of give a sort of talk in a nutshell, I'll be saying that, you know, choosing the period of the last 60 years from the time when molecular biology was established, from the time when we discovered the abundance of molecular variation, looking at that past 60 years, which is not, I don't think I can quite remember that far back, but I started really thinking about evolution in 1975 when I was studying genetics in Cambridge. I started out going to Cambridge, you know, wanting to do physics, and I wanted to do it so much, I read too much, and then it became very boring. And so then genetics, which I knew nothing about, seemed very exciting and challenging. So yeah, so I really got into the subject, what, 48 years ago, 49 years ago, something like that. So fair amount of that I can actually remember. This may make this a very over-personal and nebulous kind of talk, with certainly just my opinion on the way things are, and possibly people might not agree, but there we are. So what I'll do is really, first of all, you know, try and summarize the major advances, obviously, over this time in molecular biology, in the study of how organisms actually work, the immediate mechanism of biology, but focusing on evolutionary biology, which is, to me, quite distinct in that it's also mechanistic. It's about the evolutionary mechanisms of natural selection, random drift, mutation, and so on, but asking how populations evolve, and particularly over the last 60 years or so, the development of ideas about why they are the way they are. So we've had, I think, really big conceptual advances within evolutionary biology in understanding how we should go about thinking about these difficult questions. So, you know, I'll summarize these advances, emphasizing that actually these advances have been largely independent of each other. I think it's kind of surprising when you look at it that although if you stand back and from someone outside biology, you see this amazing gee whiz technical advance in molecular biology, which dominates the funding and the activity in biology now, the actual advances in evolutionary biology have been quite low tech and quite independent of the parallel development of molecular biology. And I'll also come back, and because I'm at heart a pessimist, really, I'll say that despite enormous conceptual progress, and despite the fact that we now have an overabundance of data, and more data than we know what to do with, we're still actually asking the same questions, in particular about the extent and the nature of selection. And if anything, they seem more daunting, actually, to answer. So, as a field, I think we're unusual in having such a clear single founder, Charles Darwin, 12th of February, 1809. Yes, we missed his birthday, but not by too much. And really, I think is the central figure who established the conceptual framework that we still work in. And indeed, that conceptual framework, I think, has been appreciated really more and more over the years. It really was not understood, maybe a century, and run into that. But much of evolutionary biology traces right the way back to The Origin of Species, the 1859 book, the abstract of his promised great work, which I'm actually glad was never published. It would have been too ponderous, I think, for us. So Darwin's ideas really fall into three parts. One, descent with modification, the idea that all living organisms descend from a single common ancestor. The idea that adaptation is driven by natural selection. And also something that Darwin put great weight on, and something he maybe discovered last of these three principles, was the principle of divergence. The idea that organisms are continually struggling, competing with each other to fill up different niches in nature, to, if you like, make a division of labour such that the diversity we see is driven by natural selection towards making efficient ways of making a living in very, very diverse ways. So the print has strangely minimised. Anyway, I never understand that. Anyway, Darwin, famously said in his autobiography that in 1830, and actually wrote in his notebooks in 1838, I think, that at last I have a theory to work. He'd understood these three principles and he had a theory to work by. And of course, he then spent, 20 years really assembling evidence to convince people that these principles apply. And I just put that sort of phrase in there to emphasize that Darwin really was a very strong theoretician. He was very uneasy with mathematics and really quite alarmed by it. But he had a very strong verbal theory underlying all of his enormously diverse collection of data. And another phrase that comes to mind is that the origin of species was one long argument. It's really structured very powerfully to making the case for his theory of evolution. So immediately after the publication of the origin, it very widely read, and the principle of descent with modification was what we now call evolution, descent from a common ancestor. That was accepted quite widely, quite quickly, surprisingly quickly. Perhaps in part because it gave people a theory to work by, a research program. It meant that many, many different aspects of biology could be brought together under an evolutionary framework. And that actually always is a powerful motivation, even if people are uneasy about exactly whether they agree with this or that part of the theory. Having a research program is enormously influential in promoting a field. But the idea that natural selection was the central process, which was absolutely crucial to Darwin's thinking, was not accepted for a long time, for many reasons, partly because it's just really quite hard intuitively to understand that the extraordinary diversity of complex adaptations that we see is driven by what was called by Herschel the higgledy-piggledy process of natural selection. Just is hard to grasp. Actually, it does work. We'll hopefully understand that, but it's not something that is immediately obvious. And secondly, though, and something that Darwin felt very much, was that he didn't understand how inheritance worked. And in particular, the theory of inheritance at the time, the dominant theory of blending inheritance, would reduce the variance in a population by 50% every generation. And this was actually shown in one of the very earliest kind of population genetic calculations, if you like, in the 1860s, in several papers, or two papers really, which showed, yes, under blending inheritance, diversity would decrease very quickly. And Darwin simply said, well, we see variation, it must come from somewhere. But this made it very, very hard to actually sustain or develop the theory of adaptation by natural selection. So, in fact, Mendelian genetics saved the theory, but this wasn't appreciated at the time. When Mendel's work was rediscovered in 1900, there was an extraordinary burst of activity over more or less a decade, from 1900 to 1910. The principles of classical genetics were established, linkage, correspondence, the idea that genes were arranged on chromosomes in a linear map, etc. All of this classical genetics was established in a very exciting phase. But the new geneticists were rather dismissive of the old Darwinian ideas and the old research program of biometry, which had been established by Francis Galton and developed by Carl Pearson and Ralph Weldon. This idea that small variations, continuous variations, were responsible for adaptations, that was seen as just irrelevant, incompatible, uninteresting. And it took into the 1920s and even beyond for the two ideas to be reconciled. And this was done by Fisher, Haldane, and Wright in developing population genetics and in applying population genetics then to many other fields, to what's been called the evolutionary synthesis, or sometimes the modern synthesis, which by the 1940s had really become established as a synthesis of Mendelian genetics with Darwinian natural selection. But I think one can say that although this synthesis did emphasize the importance of natural selection, it was rather, if you like, uninquisitive or imprecise in thinking about exactly how that worked. And there were a lot of ideas of evolution for the good of the species, about various kinds of group selection, which was losing sight of Darwin's really consistent emphasis on understanding evolution as a process of competition amongst individuals. So this is the story up to, the 1940s, 1950s. And it's probably, hopefully, a familiar story to many of you. So what I'll do in the talk really is focus on the following 60 years or so, from around the time, in fact, when molecular biology was established. There's a wonderful account of that, which I hope many of you will read, by Horace Freeland Johnson, The Eighth Day of Creation, interviews and detailed stories about how, more or less from the time of the discovery by Watson and Crick of the structure of DNA and therefore the mechanism of genetic inheritance, of the discovery by Jacob and Monod and others of principles of gene regulation, of the principles of structural biology, of the idea that macromolecules have a precise structure, it's a flexible structure, the principle of allostery, which allows a change at one site in a protein to transmit a signal to another site in the protein, allowing more or less arbitrary functions to be connected through this specific structure of a macromolecule. All these principles, which really are the key principles of molecular biology now, that was all established within about 10 years. And I put, a very rough timescale to about the time the genetic code was established and the principles of gene regulation established in the early 60s. And at the same time, more or less, or slightly afterwards, our field was really changed dramatically by the discovery of extensive molecular variation, most important by a paper by Lewontin and Hubby on Drosophila. At the same time, Harris in humans. They both discovered, using the method of gel electrophoresis, that there was far more variation at the molecular level than people had imagined. Up to that time, there were many, many detailed studies of very particular, peculiar systems, banding in snails, butterfly wing patterns, and so on. Still important systems for us now, but very hard to assess the level of variation overall from those particular model systems. I just want to say here that these two events, although coinciding in time, are kind of unconnected, because enzyme electrophoresis is really quite low tech. It depended on really specific stains for specific enzyme reactions, which required biochemistry, but that wasn't new biochemistry. That was biochemistry that was well established and maybe took a certain amount of ingenuity to get it to work consistently and cheaply and easily. But this was really independent of the developments in molecular biology. So in evolutionary biology, there was immediately a vigorous debate triggered by Kimura and also King and Duke's proposal in 1968 of the neutral theory. The idea that this molecular variation was by and large random in a certain sense. Random in the sense that substitutions between species were established by the random fixation of mutations, regardless of their selective effect, which was seen as trivial. And variation within populations was a transient phase of mutations either being lost or being fixed. So the neutral theory, strictly speaking, is not neutral. It's not saying that there's no selection. It was completely clear that adaptation is driven by natural selection. But it's simply that at the molecular level, most of the stuff we see is kind of more or less random, that only a very, very small fraction of that variation is driven by selection. And in particular, polymorphism is effectively neutral. There's also, of course, no denial that organisms are maintained by what we could call purifying selection, by selection for a particular genotype. And this is sometimes misunderstood, but the neutral theory was really setting out an argument about the nature of molecular variation, not phenotypic variation. And so the motivation for this was really twofold. One was the sort of sense that it was simply impossible for natural selection to maintain variation by balancing selection on many, many sites. And it was impossible for it to drive very large numbers of substitutions. And in fact, those arguments, various kinds of genetic load arguments, have not stood up very well because one can find ways around them. And it's surprisingly hard to come up with a strict bound on just how much can natural selection do for an organism with limited reproductive capacity. The second argument is actually held up a bit better, I think, and that is that the rate of substitution along separate lineages is approximately the mutation rate. And that's really quite striking. There's no reason under a selective model why the rate of substitution should have anything to do with the mutation rate. And yet it's a very robust result under the neutral theory. And of course, we know now that the substitution rate is not constant. There is more variance in rate than you would expect from a pure neutrality. And that actually implies perhaps some kind of selection. But overall, the order of magnitude rate is somewhat constant, is at least for any particular molecule, and it is of the order of the mutation rate. Okay. So these two arguments, I think, actually, do hold up, although it's very difficult to pin them down. At any rate, there was an immediate, very fierce debate between both of the community who had just achieved this evolutionary synthesis with its emphasis, its rediscovery, if you like, of the importance of natural selection, and resting on the many studies of melanism in moths and so on and so forth, which showed very strong selection on various polymorphisms and demonstrated balancing selection, maintaining variation. And so there was a lot of resistance to the idea that actually most of the new stuff we were seeing was neutral. But what this drove, actually, regardless of the pros and cons of the different arguments, was a development of a different attitude to studying evolutionary biology, rather than studying particular systems as idiosyncratic examples of how selection works, was rather trying to take large data sets. I mean, they were seen as large in those times, they seem small to us now, but take, let's say, data sets on the heterozygosity, on the frequencies of different alleles as determined by Alzheimer electrophoresis, and to make inferences from those using neutrality as a null model. So even if you thought that selection was really important, you would still take neutrality as your null model. So there was the development, really for the first time, of statistical methods for rejecting a neutral null. And that's really how we work nowadays. And it immediately became clear that one could just reject the standard neutral model. It just doesn't fit, because the neutral model is a very strict thing. It simply says that really the only parameter determining the distribution of variation in a population is n mu, the population size or effective population size, times the mutation rate. And the only parameter determining the rate of divergence is mu. And one can easily reject this. It's a very, very rigid model. So, for example, what's become known as Lewontin's paradox argued, this was an argument Lewontin made in his book in 1974, Lewontin's paradox is that very abundant species do not have excessively high diversity. To put it another way, under the neutral theory, the amount of genetic diversity in a population should depend on a single parameter, n times mu. And although that does increase with population size, not that it's very easy to measure effective population size, but and the genetic diversity does increase with population size, it increases only over maybe 1 or 2 orders of magnitude over the whole range of organisms, which vary over many, many orders of magnitude in population size. However, the way people resolved these, or tried to resolve these failures of the neutral model, the strict neutral model as proposed initially by Kimura, they fell into 2 camps. And in fact, this is still the puzzle we have today. So firstly, you could say, well, it's all because the population structure is more complicated than a single well-mixed population. You have population subdivision, you have population bottlenecks. So for example, genetic diversity in very, very large populations may actually be dominated by bottlenecks in the past, by speciation events which reduce diversity. We really don't know what the population structure was in the past. The second alternative, which actually was proposed by Maynard Smith and Hay very soon after the introduction of the neutral theory in 1974, was that actually in a very large population, diversity is limited not by population size, by random drift, but rather by linked selection. In particular, in their model, by selective sweeps, by the idea that a single mutation comes into the population, sweeps to fixation, and carries with it a random chunk of genome, which fixes and therefore eliminates diversity. So populations will always be losing diversity due to the substitution of adaptive alleles. And so in a sufficiently large population, the pattern we see will be dominated by this kind of linked selection. And so we still have this dichotomy, which is extremely hard to distinguish, even with the abundance of data that we have now. Okay, so in parallel then with these developments in population genetics were extraordinary technical advances in molecular biology. And I'm going to be brave and hope there are not too many molecular people in the audience and say that actually there were really not many conceptual advances since the early days of Watson, Crick, Jacob, Monod, and so on. Because you can build an organism with the principles established in the mid-60s. And of course, there have been extraordinary wealth of detailed discoveries about how, in fact, genes are regulated and all sorts of different mechanisms can be co-opted to alter the expression of genes. And there have been some really perhaps unexpected discoveries, for example, that RNA can act as an enzyme, that RNA can have functions in the organism independent of coding for protein, although Crick in particular emphasized the importance of RNA really in the early days. But by and large, the dramatic advances in molecular biology have come from ways of manipulating DNA, of cutting it with restriction enzymes, amplifying it with the PCR reaction, sequencing DNA, the development of extraordinary high throughput methods for sequencing, much of this then driven by the Human Genome Project, which put an extraordinary amount of money into sequencing the human genome. And now we have an extraordinary abundance of sequence data. Now those have the terrible shrinkage of the font. So we now have whole genomes from whole populations. And when we analyze these data, we rely on the neutral theory. An elaboration of the neutral theory, though, to allow for population structure, to allow for mate selection. So the framework we have is based on the coalescent process, which is really is a new theoretical idea that came in the 1990s, stimulated by the fact that once you get sequence data, the natural way to think about things is not allele frequencies, because every chunk of sequence is different from every other chunk, even a reasonably short, 1,000 bases. There'll be many, many combinations typically. Instead, you think in terms of their relationship. And if you see a set of sequences, you can immediately infer, at least approximately, their genealogical relationship, which you have a tree-like structure, and you need a theory which thinks in terms of these trees. And the coalescent process is a very simple model that says that if we take two genes and trace their ancestral lineages back in a single population that's well mixed, the rate at which they coalesce in a common ancestor is 1 over the number of genes in the population. And it's very simple. In any generation, there are these two lineages looking for parents. There's a chance 1 over the number of genes in the population, 1 over 2 NE, that they'll be in the same parent. So that's a beautifully simple model that reproduces many of the theoretical results that were already known from Kimura's diffusion theory, but in a much more elegant way. But it's clearly not enough to explain the whole rich variety of models that we want to look at. And so one thinks now of the structured coalescence, which has lineages can go back in time and they can move from place to place by migration. They can move from genetic background to genetic background by recombination. And so we can actually understand neutral variation, even when it's embedded in a background that is structured in space and that is structured by genetic diversity at selected loci. So that's a very powerful framework that can, allow us to predict in great detail the properties of samples of genes. So evolutionary genomics has really returned to what I'll call the classical view. This is a term introduced by Lewontin in his book. Again, Lewontin contrasted what he called the classical view, which is a very simple one, which is there's good and there's bad. There's in any particular environment, there's a particular DNA sequence, a particular gene that gets best, and the variation around that is due to deleterious mutation. And that populations evolve by occasional substitutions. When the environment changes, some mutations will become favorable, they will fix in the population, and move to a new state. So this classical view is a very straightforward view, and it is actually the view that dominates analysis of sequence variation, in that we typically think either in terms of what's called background selection, in which selection against deleterious mutations affects neutral variation, or we think in terms of positive selective sweeps, and we try and infer those. So this classical view contrasts with what was associated particularly with Dobzhansky, who was, more entranced by variation. In particular, he studied Drosophila inversion polymorphisms, where clearly different combinations of genes were maintained in the population by some kind of balancing selection. And he saw variation as essential to adaptation and as being maintained by selection. And so really the main viewpoint of evolutionary genomics now is dominated by the classical view. And to me, it's a rather sad, dull view. There's something gone terribly wrong with my slide, but anyway, there's nothing really worth reading. So anyway, this view, this classical view, is all very well, but it implies that there's a limit to how much variance in fitness there can be, a limit how much selection can be active. If we think about variance in fitness, variance in numbers of offspring, it's quite a large value. Some of us have more children than others. And that's something tangible to measure, but measuring the heritability of that is difficult. And yet, at least my feeling, it's surprisingly hard and rather embarrassing that we haven't really pinned down good estimates of the genetic variance of fitness, but still, most quantitative traits, even those that affect fitness, have a high additive genetic variance, suggesting that fitness itself has a substantial additive variance. And yet, under the classical view, the fitness variance is limited by the mutation rate. Actually, the prediction is fitness variance is total mutation rate times selection coefficient, which can't be more than a few percent, much less perhaps. And it's limited by the number of substitutions. It's actually limited by the rate of substitution times the selection coefficient driving that substitution. Again, probably a rather small value. So the whole viewpoint is neglecting balancing or fluctuating selection. Okay, so I don't want to go on for too long. I will be thrown out by Ashley, I think, soon. So I'll try and whiz through, because I hope everyone will accept the wonderful achievements of evolutionary biology over the last 60 years. And I'm really not going to elaborate. Oh, I can't elaborate. It's getting smaller and smaller. It's amazing, the power of PowerPoint. I think it's, I don't know what to blame really, probably myself. Anyway, I will just go on to the next. Okay, I will list the achievements, which are, phylogenetics, the ability to estimate phylogenies, to interpret them, to apply the comparative method, to ask whether there's a significant association between different traits on a phylogeny. Extraordinary work on speciation, the genetics of speciation, actually driven by people like Jerry Coyne, Alan Orr, and others in Drosophila using classical methods actually to really pin down the genetic basis of incompatibilities between species. The resolution explanation of Haldane's rule and the large X effect and so on. The evolution of sex and recombination, understanding at least theoretically why it is that sex and recombination can be favoured, because they facilitate the actual action of natural selection. Kin selection, the understanding of how selection can work at different levels, how it is we can understand group selection, kin selection in maybe different but parallel ways. And also ideas introduced by Maynard Smith and Satmari in thinking about how complex organisms evolve through the combination, the symbiosis, the cooperation between simpler reproducing units. Development of theories of population structure, ecology and evolution. And then most recently, perhaps, quantitative genetics, genome-wide association studies and so on. Until we get to that point, the whole list of achievements that I was talking about were actually largely independent of the developments in molecular biology. Maybe phylogenetics, so of course we've got much more data to do phylogenetics with, but actually the methodologies can be applied, the logic can be applied independent of that. And certainly work on evolution of sex, speciation, kin selection and so on, I think doesn't depend strongly on the sort of technical advances. In fact, this work was developing during the 80s and 90s before we had an abundance of sequence data. But now we are in the realm of being overwhelmed by sequence data. We have whole genomes, potentially whole populations. I think 10% of the population of Estonia has been sequenced. Massive data sets. And that's actually revealed something that, in a sense, we knew all along. Darwin perhaps would have known if he'd understood genetics, but maybe that's a bit of a stretch. That most of the traits we care about are highly polygenic, depending on an extraordinary number of variants of small effect. And the reason that these enormous sequencing studies have been done in humans is because small studies found a few genes which explain a small fraction of variants. So then you do a bigger study, bigger study, bigger study, and you discover that as you increase the size of the study, you get an ever-increasing fraction of variants explained, implying that we're actually close to the infinitesimal limit, the limit where actually traits variation is driven by very small changes, very small biases away from neutrality at very many loci, which actually is implied and was in some sense understood in the artificial selection world, simply from the observation that if we select on a population, it will keep changing. It will change by 10 standard deviations over 100 generations. It will keep moving. And that implies there has to be lots of standard variation released by recombination. Okay, so I'm coming to an end, I hope. Yeah. So evolutionary biology has advanced in many different directions, advanced conceptually and empirically over the last 60 years. And yet, particularly for someone like me who grew up in the middle of the neutralist selectionist debate, no, yeah, okay. Why are some things big and some things small? It's very odd. Anyway, it doesn't really matter. We haven't addressed, actually it's getting smaller and smaller. Like Alice through the looking glass, yeah. So we haven't answered the original question. The question's about the nature and extent of selection. What the neutralist selectionist debate was about was what fraction of divergence and polymorphism is driven by selection as opposed to drift. And although we have an abundance of candidates where we do a genome scan, we find some signal of something going on, something exciting, we have a few model systems where you can really investigate those, but in most cases we're left with peaks in a genome scan. And we don't have any way actually of saying, firstly, really knowing if those are real or if they're false positives driven by demographic fluctuations, or knowing if they are real, what was the strength of selection? 1%, 10%? We don't know. And so I will finish before the font shrieks. So I think another thing I want to say, having given this sort of over-rapid overview of the whole field over 60 years, but okay, so the main point I wanted to get from all that is that yes, evolutionary biology has advanced. Yes, we benefited enormously from the technical advances in molecular biology. We have a wealth of data, but actually those data are the most problematic part of evolutionary biology now. We really don't know what to do with them and how to use them to answer the original questions. But beyond that, I think it's interesting if you had been able to read the different topics, into which the field is fragmented, you will see that we've lived in very different worlds now. The world of phylogenetics lives in a different world of conferences and so on. Even within phylogenetics, there are people trying to estimate phylogenies, people trying to interpret them in various different ways. There must be dozens and dozens of different subfields within evolutionary biology. And I find it disturbing in a way that most people coming in don't have a kind of overview, I think, because it's a subject that isn't taught in the early stages. Physics is taught in high school. You come into university to do physics knowing about the basics of differential equations and about waves and about electromagnetism and about the quantitative basis of the subject. And yet you would possibly never learn the quantitative basis of the subject in evolutionary biology. Typically, you would start to learn it when you start a master's or a PhD. And so it's a very strange situation for the subject, and I'm not sure what we can do about it. Anyway, on that possibly pessimistic note, I'll end. Yeah.
Thank you so much, Nick. That was great. We don't ask questions right now because we're saving them for the debate. Remember them, please don't forget them, because now we'll have a very interesting talk coming up. I know it's difficult. He's talking about balanced selection. I cannot ask a question. It's very difficult, but just keep them for later. So our next speaker is Ashley Griffin, who is going to talk us about something slightly different, which is social evolution, I imagine. So she's done some amazing work on the evolution of cooperative behavior and understanding social evolution in a range of different species. She studied, apparently, Nick was one of her lecturers in Edinburgh. She got a PhD studying social evolution in meerkats, which is really interesting. Then she became a Royal Society fellow, research fellow, and she's a professor at Oxford now. I'm sorry I cannot give an introduction like the one that Richard gave to Nick, because I don't know her that well. Maybe in 20 years when we invite you again, or the next CEE directors invite you again, then maybe they can do that. Thank you so much for agreeing to talk.
Yeah, my list isn't quite as long as Nick's list. Maybe in 20 years' time. Maybe in 20 years' time. So I'm actually, so I will talk about some social evolution, but I really sort of wanted to take this chance to have a slightly more broader view of actually some of the themes that Nick has introduced so beautifully in his talk, about how we as evolutionary biologists, our field, fits in with some of the more technology-driven advances in the life sciences more generally. So maybe sort of thinking a little bit about the questions that Nick's raised about how we integrate a little better with some of those fields, which we all know get way more funding than we do. So I'm going to start a focus on a sort of a really practical consideration, and that is how to watch things. Darwin's methods really centered on meticulous observation, and so this is like a example of a etching that he made himself of the Galapagos finches. And you can see like the beautiful, like meticulous detail that he's gone into in these drawings. And this is what boils down to like the method of understanding the adaptation in biology, where we make these observations and note variation and come up with hypotheses about how to explain those variations. So observation is very much at the starting point of what we do. And that's sort of like so that's how do we go about doing that first step when we're confronted with something like this? Okay, how do we work? How do we make a start in understanding the adaptive processes that are driving organisms in a habitat like this? Most of the organisms here are invisible to the naked eye, although I did spot there is a little worm there. Okay. Okay, and these are plant roots. But there, there's a complex ecosystem there which we can't see. So it's how do we go about thinking about adaptation of this invisible world? One of the strategies that we have, the main strategy that we have probably, is to just sample from these environments and take them back to the lab and try to grow what we find there. And we as in microbiology, we work with what we call isolates, and they're called isolates because we isolate them from the selective environment from which we found them. They're isolated from all the other organisms that they interact with in their natural habitat. So we might be able to sample and get hold of some isolates from something like this. We might recognize that by doing that we are missing out masses of life that are unculturable. So we might sequence it and see find out at least what's there by doing that. But then that information doesn't tell us anything about how they interact with one another or abundance or so on. We might try to recreate an environment like this in the lab and try to have like a little mesocosm of a soil environment inside the lab, and and that that's maybe we get so far with some questions with that, but it's always like a little bit of a wing and a prayer. You know, you don't quite know if you're capturing the selection, the selective pressures that you're interested in the lab. So, how do we go about studying adaptation in an environment like this? Okay, so at this point, I'm going to bring in Nico Tinbergen, because Nico Tinbergen was somebody who solved this problem a little bit in a completely different scenario back in the 1970s. He won the Nobel Prize along with Karl von Frisch and Konrad Lorenz for his work on animal behavior. And he had this rather sort of charming way of summarizing his achievements by saying, well, you know, we're just mere animal watchers, he said. That's what he referred to himself as. But what he did was revolutionary at the time, because most of the biology that's done on organisms in the soil, in a patch of soil, and these microbes that we were thinking about just now, are mechanistic. So there's a lot of biology going on about viruses and fungi and bacteria, but it's all very mechanistic. And that's the scenario that was going on in the animal behavior field in the 1970s when Tinbergen was thinking about behavior. So this is an experiment, this is Pavlov's dog experiment on looking at the conditional response mechanism, which I rather love. We all know about Pavlov's experiment, but this is, you don't think about how he actually did it. It's nuts. He had this dog attached to this, a very docile dog, attached to this little tube that collected its drool, and down this tube and went into this measuring cylinder there that was attached to a little recording device that record a drool recorder. But you know, like there was this study of animal behavior in the 60s, 70s was very mechanistic, like black box, sort of trying to think about how behaviors occurred rather than why. But Tinbergen, as an animal watcher, understood that if you really want to understand, you know, why a dog looks like a dog and acts like a dog, you need to go and study wolves in the forest, right? You need to understand the selective environment in which they have evolved. And the brilliance of Tinbergen and why he, you know, we still sort of teach Tinbergen's writings and when we're teaching undergraduates today is because he understood that those two approaches are complementary with one another. You know, they're not opposing with one another. And so he had this like 4 classic questions. I was at Pop Group in January, and I think it was a talk when somebody asked the audience what Tinbergen's 4 questions were, and it was woeful recall about what the 4 questions are. So I'm just going to quickly skip through. So he was thinking about like the how questions. So like the mechanism, if we want to understand a behavior, we have to think about how it works. We have to maybe think about that in a mechanistic way, but also in a developmental way. So how does the behavior fit in and how change with the developmental process of the organism, but also like the ultimate causation, the evolutionary explanation. So phylogeny, how would the behavior compare in unrelated species? How do we, you know, think about it in the context of where the organism fits in with the tree of life? And finally, function, how does the behavior impact on an animal's survival and reproduction? And this is really sort of like where this is obviously the inquiry that we make when we're applying Darwin's methods and theory of natural selection. So this part of biology is manifest in this like field behavioral ecology, which is like all the generations in this room have different versions of this book that they had when we were students. And so this is the field of behavioral ecology. And this is really the field where we're like sort of where biologists are asking functional questions. So it's number 4 on Tinbergen's list, asking like why things are the way they are. And as Nick was saying, this is a very niche when we think of like all the life science departments in our universities, the funding that is distributed among the life sciences, the functional sort of explanation of question number 4 is taking part in a really limited number, by people in this room. So that's us. We're doing that. But there might be some other fields that think they've done evolution, because they've and I think we all laugh because you know what I'm talking about. But actually they're really doing much more descriptive stuff, like, so it's really much more like question number 3, which is just to describe evolutionary processes and building trees. They're really not doing really not trying to make functional explanations for the behaviours that they are observing. Okay, so I was asked to sort of give a sort of a pitch for where we might be going in the next 30 years. I have no idea, but this is what I've got. Okay, so I'm going to sort of I'm going to try and suggest ways. I'm going to try and be positive, and I'm going to try and suggest ways that we can bridge the gap, as Nick was saying. The two fields have been evolving in parallel. How can we sort of integrate them more effectively than we're doing? And to try and explain that, I'm going to use. I've got a case study, and the case study is me. Except, weirdly, there was supposed to be a picture of me feeding meerkats on that slide, and it's gone. So it's just me. That's embarrassing. But anyway, so let's have some meerkats up without me. But I started doing my PhD on meerkats. These are cooperative breeders that live in the Kalahari Desert in South Africa. And I was, my job in that project is with Tim Clutton-Brock in Cambridge and Josephine Pemberton in Edinburgh. And my job in that project was to genotype meerkats and find microsatellites and measure relatedness between meerkats. And it was really a lot of fun. So I went here, and that's the photograph I was going to show you, was me in the Kalahari looking intrepid. That was only for a few weeks. Most of the time I was in a basement in Edinburgh doing PCR reactions, a lot of them. So the idea of this project was to investigate the evolution of social behaviour and trying to test some theories about kin selection, about why social behaviours have evolved in a species. Basically, they encapsulate the problem of social behaviour beautifully, because you have like a dominant pair in a extended family group like this one that produce all of the offspring, but the care for that offspring and all the cost of the care of the offspring is paid by these non-breeding adults. So the question is like, why do the helpers help? And it sort of encapsulates this problem of social evolution more generally, of like, why should you pay a cost to help another individual instead of putting that into your own reproductive success? I'm not going to talk more about meerkats. When I sort of come to the end of my PhD, okay, I got pregnant and I didn't have a job and I hadn't published any papers. So it did take me a while to get started again. But when I started again, because I'd done my PhD in Edinburgh with people like Nick and others and Charlesworth around, I was really thinking always about population genetics. And I was reading the papers, the theory papers about from Taylor and Frank and people like that. And I wanted to sort of test, there was no sort of real empirical test of those papers at the time. It was very much just theoretical. So I wanted to move from meerkats to a system where I could do experiments. So that's why I sort of switched to bacteria, because I wanted to do experiments on them. This is Pseudomonas aeruginosa, and I worked with a trait where the cooperators were green and the potential cheats that didn't perform a cooperative behaviour were white. That's all I knew about bacteria at that time when I started. But the important thing is you could see whether a cell was a colony was a cooperator or not. My font is shrinking as well. So I thought it wasn't going to happen to me, but it has happened. So this is so cooperation in bacteria is a little different from meerkats, but it's essentially the same thing. Individuals like this cell here are paying a cost to produce this compound, which it releases into the environment, and it forms a benefit. And all the cells benefit. So the white ones that don't produce this compound have a disadvantage because they're not paying the cost of producing it. They're enjoying the benefit. And that's basically a sort of like, that's how the main way that bacteria have cooperative interactions with one another. So I did all of these experiments to test theories about the evolution of cooperation using my cooperator and my mutant, my non-producing mutant, for this molecule called pyverdin, which is a chelator of iron. Iron is a limiting element in when a bacteria is infecting a host, because our iron is like bound up really, really tightly in hemoglobin. And so it needs to do something to grab that iron. And what it does is it makes this stuff called pyverdin, which is green. So I could mix these together under different circumstances, and test models about density dependence, frequency dependence, all of that, and count them on agar plates. So it's a really neat system for testing theories about, what's the effect of relatedness on the proportion of cooperators, do the selection experiments and test all these different things. Okay. So it was okay. This is all very well. Like, I tested a lot of different theories using this my experimental setup without really thinking about bacteria. But eventually, I did actually start to realize that they were organisms, and that maybe it would be interesting to think about social behavior, the evolution of social behavior in bacteria. Okay, because these experiments are very much like this kind of scenario, the equivalent of this in meerkats. The bacteria that I was working with in these experimental evolution experiments are like taking meerkats into the zoo and watching what they do there, and trying to interpret their behavior as in terms of adaptive benefits, which of course is very difficult to do. Whereas from like meerkats, we can go into the Kalahari where they live, where they have evolved, and we can, as humans, even though we're not meerkats, we can kind of get a grip on what it's like to be a meerkat. We can go there. The same rules of physics are applying to us as apply to meerkats. And we can understand that it's hot. We can understand that it's very exposed. So, the selection pressures are, we can kind of make some sensible guesses about what selection pressures are, at least. But how do we do that with something like a bacteria that lives in somebody's lung? How do we know what the selection pressures are? How acting on a bacteria living inside somebody's lung? Here is like an infection here. And so for the next sort of phase, and all of this stuff is funded by the Royal Society. I would not be sitting here if it wasn't for the Royal Society. So if there is anybody from the Royal Society here, thanks very much. Nobody would fund it, only the Royal Society. So what I want to do, the next phase, I really want to understand, can we explain do these cooperator cheat dynamics that we can see in our experiments also happen in nature? Can we think about adaptation of social behavior in bacteria? So the first thing we wanted to know is just look at what the pyverdin production did in infections, in these lung infections. And so with collaborators in Denmark, we had lots and lots of samples of longitudinal sampling of people with cystic fibrosis, of these long-term infections. We could, sample bacteria over many, many years, many patients, and ask, what happens? Do we see any evidence for these cheats being able to invade like we see in our experiments? And these were all done by Sandra Anderson, who's a postdoc in my lab at the time, and now she's a PI in Copenhagen. And this is what we found. So first thing, yuck, right? This is not a lab experiment, not a nice lab experiment. This is what happens when you just take bacteria out of sputa and then plot what it does, okay? We did see that it has this is time along the x-axis, and the pyverdin seems to be declining with time. If we pick apart the pattern a little bit, it's a little clearer. So Sandra said, well, she's interested in these strains that produce very little to no pyverdin. They're really not making any. So she categorized, like she had a cutoff and called a certain type of low producers, ones that produce less than 1% of the average. And then she finds that these very low producers increase over time. So as the infection gets longer, the percentage of non-producers goes up and up and up. So there seems to be like a sort of the pyverdin, which on the face of it, everybody thought that's a good, that's something bacteria make to get iron in the host. They don't seem to be maintaining it in a natural infection. So the question is like, and this gets us back to like, us thinking about adaptations. What are the selection pressures driving that decline? Okay, it could be that they're out competing cooperators, they're exploiting cooperators, the pyverdin producers, and spreading. Okay, that's the sort of classic sort of cooperator cheat dynamics thing, which I'm representing with the meerkat. Okay, but the alternative, perhaps even simpler explanation, is that they're just losing those traits because of redundancy. So maybe the benefit of producing pyverdin declines over time because there's more iron, free iron around as lung tissue degrades. Others maybe alternative strategies. So the challenge is how do we know? How do you tell what selection pressure is acting inside somebody's lung? Yeah, how do you go about this? So that's what the sort of like, I'm going to be trying to be positive and give a positive message by telling you that there is ways, right? There's ways, okay? And the first way that you can do it is that you can look for clues, right? So we don't know what it's like in there, but we have access to information that tells us what selection pressures are being experienced in there, what's important to bacteria in the lung, and that is through the genome. The genome is like wet sand, if you like, and you can see the footprints of selective pressures that they're experiencing in the genome. So happily, the molecular biologists who, as we know, are going off in parallel, doing other things in parallel, those wonderful molecular biologists had worked out the genetic architecture of the trait of pyverdin production. And this is a diagram of it here. So we have a bunch of genes that are involved with producing the pyverdin molecule. So they synthesize it. It's released through the membrane into the environment where it can do its thing. There are other genes in the system that are responsible for producing the receptors that sit on the membrane and take in those pyverdin iron complexes back into the cell where it could be used for metabolism. So there's some genes that are paying the cost to make the thing, and there's other genes that are paying the cost to take the pyverdin back in. And we thought that if those non-producers are cheats, they would have mutations in their genes for synthesis, but they sure as hell would grab on and hold on to the receptors, right? Because they're being a cheat is all about take and no give, okay? So that's what we did. And the really great thing about this amazing study from my collaborators, Søren Melin and Hella Krogh-Johansen in Copenhagen, is that it's so huge. There's lots and lots of different patients. Some of these patients have lung infections where the pyverdin producers are there, and others have lungs where they've all been lost. So we can have some people with pyverdin around in the lung and others without. And we can ask, where do we see the mutations, okay? Do we see a difference in the number of mutations in the receptor genes between these two groups? And the answer is that we see a very, very marked difference. We see, so the blue here is where the pyverdin is absent. The grey you can hardly see because they don't have any mutations, hardly any mutations at all. They're holding on to the receptors. But as soon as the pyverdin is absent, it disappears. So if you're lucky enough to have the information about the genetic architecture, and you have a really good think about what of really trying to be careful about the hypothesis that you ask, then the wealth of data that we have that Nick was talking about the wealth of data is like you guys all have the expertise to use all that data to ask questions like this, okay? A microbiologist would never have come up with this, okay? It needs our expertise to do something like this. So the second strategy is maybe an obvious one because it's, you know, it's been around a long time, is in finding new ways of seeing really small things. This is a picture of a louse taken from Hooke's micrographia from 1665 after the invention of the microscope. So we've been at this a long time, trying to see smaller and smaller things. I really love this one. Because of the hair, like that he's thought to have it actually holding onto the hair so you can get a feel for, you know, what it looks like in your head. So anyway, so we can find new ways of seeing, and we've done that. And for example, we can look to all the amazing molecular biologists can construct the structure of the receptor that sits in the membrane. And they can tell us exactly where in that structure the mutations that are changing the amino acids. And so all these mutations here are happening in this in the lid that shuts and opens, whether it wants to receive pyoversion or it doesn't. So it's we can make testable hypotheses about molecules like this one in the same way that we can that Darwin did about the shape of a finch's beak, you know, in the Galapagos. Now, you guys can do this, right? But molecular biologists don't necessarily think about asking the same kinds of questions that we do. This is completely different. So this is now a sort of another way that we can, another scenario in which we can think about adaptation of things we can't necessarily see with the naked eye. Isn't just individual organisms, but it's also cells within an organism. So we can think about adaptation at different levels if we have these new ways of seeing. So this is a planarian flatworm. If you chop them up, they grow into lots of little worms, like they're real sort of like weird alien things. And we can now, with this is an in situ hybridization where all the stem cells, the totipotent stem cells, which can be used to make new eyes, new gut, new nerves, are stained in yellow. And so we can see that the amazing regenerative abilities of something like this flatworm, we can start to understand it by looking at images like this. Yeah, so we don't have like stem cells like we do, just in in bone marrow. Once we're adults, all the way through their lives, they're fully totipotent. So these kinds of images can let us think about not just adaptation at the level of organisms, but also at the level of cells and cell lineages within organisms. The problem with these strategies, the in situ hybridizations, they're like the new way of sort of that we can see things in this amazing detail, is that the organisms that we're looking at are on a slide in a microscope, or they're in the lab, and again we've come back to that problem of sort of taking them out of their selective environment. So the third strategy that I think is important is trying to think of ourselves as the makers of the matrix. The makers of the matrix. What are the names of the things that have the matrix? I can't remember. The things that make the matrix. So our bacteria or cells, or they're in this simulation theory hell, right? They think they're living their lives, but actually we're watching them and we can watch what they do. And there's certain things that you can do in the lab where the entities that we're interested in explaining adaptation, they don't know that we're watching them. Okay, they just think that they're plants or cells, and I think that's something to sort of another strategy that you can think about. I really hope this works, because I wanted to show you a really amazing film which is an example of an actual matrix which is in a matrix. Okay, it's a matrix of mycelium hyphae that doesn't know it's in a lab. Okay, it thinks that it's just connecting plants. and it's work done by Toby Kears and Tom Shimizu, and I think we just watched this. I'm not gonna, it has some spooky music, which I hope is not going to play. But what I want you to see is like, this is just like, you know, people just thought this is just like, yeah, there's nutrients kind of travel along these mycelium from plant to plant. But thanks to technology, and actually for the first time being able to watch that by marking carbon atoms in this, these are like these glowing bits of carbon that are being spread around the network. Watch the way they move around. It's insane. They start going one direction and they come back the other direction. The really cool thing is they're going in two directions in the same hyphae, all of that. And these are weird organisms that, you know, they don't have a septum, so like the nuclei are moving freely. What even is an individual? So there's so many like interesting evolutionary questions about this system, but just watch this. This is the matrix in the matrix. Is it going to work? Yes, it is. Sorry about this. So I mean, that is like, you know, that's amazing, you know, like that's like, we can watch that just in the same way that we go to the Kalahari and watch meerkats now. You know, that's just insane. These are the people that that produced that, and I wanted to mention, give them a special mention here, because they're sort of like poster children for the kind of collaboration that I'm trying to suggest that we all invest more in. This is Toby Kears, who's an evolutionary biologist who's been studying fungi all her career, and this is Tom Shimizu, who's a biophysicist, and they've been working on this project together, and really successfully. And there's no way that Toby could do that work without Tom, and you know, Tom's not even, wouldn't be thinking of the same questions if it wasn't for Toby. So we need more of this. I've written that in my notes. Yes. Okay. We need more of this. And I'm going to have a little bit of a dip in. So it's all been very bit positive. So I'm going to bring in some negative. Sorry, John Welch, my negativity. I don't know if how many of you know this paper. Yeah, a few of you do. It's a brilliant paper. It's a very funny paper as well. I read it last night and I was actually laughing out loud. It's really good. But I wanted to draw your attention to it because he makes a really important point here about the fact that evolutionary biology, more than any other field, seems to have like a constant fight on its hands to sort of defend itself. And I've just picked out a little line here where he describes it quite well. One problem of natural selection is its deceptive simplicity. It's an idea that we all think we understand. The simplicity is deceptive in a second sense, that our intuitions about natural selection are often very poor. Nature is full of traits whose complexity makes it difficult to see how they can be accounted for by natural selection. And this is the really key thing. It's an idea that we all think that we understand. Nobody that's been a student of Nick thinks that, but you know, most people do think that. And I think that's like part of the problem. If we don't, what I'm trying to say here is like, if we don't fill the gap of like trying to bring evolutionary biology into these different fields, then they will fill the gap badly. Right? I think there's like an overconfidence in thinking that you understand natural selection when actually it's really hard to predict to make sensible hypotheses. It's hard to know, to think about, what's optimum behavior and do the kind of like really like meticulous and beautiful work that Darwin did on on Galapagos finches. It's hard, and if we don't all get more involved, it's going to get done more badly. And John's got John's makes that point really well. So just an end note, I wanted to finish with a quote from Darwin, and I found this one of him. This is him actually in the Natural History Museum. Ignorance more frequently begets confidence than knowledge. And I think that's what I wanted to finish on that note, because we all, the more that you know about evolutionary biology, I think the less confident you become. And I think that's really healthy. And so I would strongly advocate in the next 30 years that we make more of an effort to sort of join that gap and don't believe like the people when they say they're confident about what they're doing and that. Okay, thanks very much.
Thank you so much. That was fantastic. So now we're gonna have our both speakers sit here in our beautiful green chairs. I stay here. I realized I didn't introduce myself. My name is Aida Andres, and I'm a professor at UCL and also co-director of CEE. And I'll be here probably not speaking very much, but helping the debate. And now is when you can ask questions, either to one of our speakers or to both.
So questions, please.
Thank you. So for the second speaker, when you were talking about that the lung bacteria and how they were cheaters that were losing those iron-producing genes, I was just wondering how low could the proportion of producers get in the population before you get to the point where everyone dies, essentially?
That's a really good question, and when we published it, that was what one of the reviewers said, and we were kind of like, oh, we don't care, it doesn't matter, but then we actually thought, well, let's have a little look at that, and it ended up being an entirely like amazing story, which I haven't had time to tell you about today, but in brief, what they do is they switch to this completely different iron uptake mechanism, which is private. And it's just like, so they just eat the iron just comes like straight through their membrane. They only do it when they completely lose the pyroversin. They don't do it any other time. And we did some experiments to show that that's likely because iron is toxic. You have to have there's a very sort of like narrow window of like how much iron concentration is healthy. If it can be toxic at too high, and then you don't have enough, it's also bad. So the control of having just the right amount is really difficult by this mechanism, but that's all they have. And so they switch to this sort of rubbish private mechanism once they lose all the pyroversin. So it's a really interesting story, actually. I don't think so. Yeah.
Hello, great talks, both of you. Thank you. My non-science friends always ask me this question, and I'm not sure how to reply, but are humans evolving? Talking about selective pressures and stuff like that, and how to measure that, because we've got medicine and technology. So what are your thoughts on that?
Well, you mean, are they evolving now at this minute? Yes. I mean, some of the best examples are adaptation to agriculture, to living in high altitudes, things that happened relatively recently, I guess, things like lactose tolerance, living in Tibet or, highlands of Africa and so on. malaria resistance. I mean, lots of our examples of adaptation have come from relatively recent evolution in humans. I guess you're thinking about even more recent evolution. And people sometimes say, oh, evolution's ground to a halt because everyone's happy, everyone. But no, we have different numbers of children, and that must be to some extent heritable. What direction it's going in is pretty hard to say. But yes, why should we not be evolving?
Actually, do you want to answer the question?
Oh.
I'm not really. I would have said exactly the same thing. You know, like there's we are still like exposed to different diseases all the time, and even just by drift, you know, they're still evolving, even though we're able to control the selection pressures from disease to an extent.
That's a good question.
Yeah,
Ashley, right? So I really liked your talk, and especially your idea that we need to do kind of missionary work out in other fields, and get people to think hard about what you mean by function and so on. But how do we do it? Because missionaries aren't always welcome.
Well, that's kind of the point that I was trying to make a little bit with John's paper, because he's kind of got the answer to that in that paper, and saying that they don't know that they need us. You know, there's like a sort of an overconfidence because on the surface of it, natural selection is an easy thing to do. I think like a good strategy, if I think about Tom and Toby, is just to sort of hang out with one another. You know, go to the pub with one another, have an idea. I don't know. I mean, I think it's actually really difficult. And I was speaking as somebody who's come from the coalface of all the interdisciplinary science that we're supposed to be doing. It's actually really really tough. It's really tough to even try to explain why functional questions are interesting or important. I think there's a lot. Sorry to not be more positive, but I think it's really hard. But if you can, if there are if you do have opportunities to collaborate with somebody that's like working in these different fields, then really take the initiative and really try to get involved. Is all I can suggest. Sorry.
Yeah, I mean, I think there's also a difficulty in perceived status, and often we feel the need to chase after shiny new technology, and then that's and we can do amazing things with it. But on the other hand, when you go in the other direction, as you say, they don't necessarily know that they need us, and you may end up being somewhere buried in the supplementary information, where all the exciting evolutionary biology is just some little addendum to finding the gene, Yeah. And it's very difficult to change people's values. They put value on finding the proximate mechanism and not question number 4, which we value.
it's a question. Yes, there's a question here.
Hi, very interesting talks. I wanted to ask, it's kind of a general question, but what was something that made you want to go into science in general, and then maybe something that made you want to go into specifically more of like an evolutionary aspect for both people, I suppose?
Oh, thank you. So, yeah, I was always interested in science and making things and understanding stuff, and excited more about physics than about biology. And I got into evolutionary biology. It's a bit embarrassing actually, because even though I collected fossils as a kid, I never really thought about it. I thought a lot about kind of physics and all that stuff. But when I got into university, and it was a great thing in Cambridge that they still have a natural sciences degree, you had to do some biology, and it was, of course, completely new to me, and so seemed much more exciting. I was attracted by the universality of Mendel's rules and about the sort of big questions, and the amazement that you could apply natural selection to explain so many things. So it was kind of luck, a lot of it.
I've got an utterly dull answer to that. I just I always wanted to be a biologist. It was dull. But going into evolutionary biology, and I don't want to embarrass you, Nick, but being an undergraduate at Edinburgh, it was a really, really exciting place to be an evolutionary biologist in sort of early 90s. when I was the student there. You know, we had so many, you know, brilliant people from all over the world coming, Sally Otto and Mike Whitlock, Mark Kirkpatrick. I took it for granted. I just thought that's what it was like. And then ever since then, I was pining after those times. But it was just a brilliant place to be, just at the right time. And so, you know, that was really what got me excited about being any kind of evolution. Didn't really care which kind it was. Yeah.
Thanks. I have a question about selection on genome architecture. So on one hand, we have some evidence that there might be selection on how big genome is. On the other hand, we have some evidence for selection on how complex traits are and how complex their genetic architecture is. So for example, selection for plasticity. And so for UNIC, I wanted to ask you whether you have any thoughts, how does that sit with people who are proponents of purely neutral evolution? And for you, Ashley, I wanted to ask if you have any thoughts about potential function in evolution of such traits.
Traits like genome size, you're saying? Or yeah, okay. Yeah. So firstly, I don't think there's anyone who's a proponent of purely neutral evolution. We'd all be a kind of sludge, you know, if it was purely neutral. But what is difficult to pin down is the idea that, you know, as I find plausible that most of DNA sequence is random, is neutral, or very close to neutrality, and yet selection can push it gently in this way and that. And at the trait level, things look very much under the control of selection. So, I mean, things like selection for genome size, we know that, you know, it is selected because plants with different life history, animals with different life history will have different genome sizes. There's a kind of general pattern. And yet, of course, whether you have this or that base pair, one or more or less, doesn't matter so much. But, you know, as I was trying to explain not very well was, you know, how do we get at these questions about what fraction of molecular function is really functional in our strict sense? Yeah, I think it's hard. You got it.
Well, I was thinking about it. It's like your questions are really interesting. It's a really great question. I was it made me think about in microbiology. We think about genome size between different species of bacteria quite a lot, and it's a really good example of a question that's staring everybody in the face, but it's kind of been neglected. I think because people tend to study just one species. If you're interested in mechanism, you're a Staphylococcus person, or you're a Pseudomonas person, or an E. coli person, because the mechanisms differ between species. So you might spend years and years just doing working the same species and going to the conferences where everybody's talking about the same species. But it's a question that really requires a comparative approach. And that's something that people are only really starting to do now, in at least in microbiology. So I think those kind of questions that you're talking about are really interesting, that are really gettable using a comparative approach.
It's a really good point, because if you look at kind of the natural history that I guess most of us are most familiar with, we're really interested in the whole range of life that we can see anyway, and comparing them and valuing the diversity. Whereas if you study, you know, these molecular phenotypes, and there's really interesting molecular natural history, it's very hard to get a sense of the broad sweep, especially when we think of, let's say, single-celled eukaryotes, which is most of them, and we know very little about them, certainly at Merck.
Can I just follow up with Raphael's question on about trade-offs, or kind of about genome evolution? Because Nick, you present this idea that most selection is polygenic, most traits are polygenic, and lots of genes are controlling them, and it gives us a kind of idea that phenotypic space is fairly free. You can move in phenotypic space quite easily, and yet when we look at organisms, we see quite persistent trade-offs across taxa. I mean, where are they coming from? If the genome is so sort of labile and able to evolve in these new directions, are they all driven by ecological interactions? But
there have to be trade-offs just through physical constraints. There are limits on what can be done, however wonderful organisms are. So you can have a freedom in DNA sequence. There can be a vast range of sequence space which can achieve a phenotypic result in many different ways, but those phenotypic results are constrained by trade-offs. It is difficult. I find it difficult to pin down exactly what we mean by a trade-off. What's the difference between a physical constraint and a more evolutionary constraint? Because organisms find ingenious ways to get around the physical constraints. So yeah, it's tricky.
Thank you. Go back to the audience. More questions, please.
Great. Thank you for the really interesting talks. I guess everyone says this, but I think this is really an ignorant question. But what is the topic of the debate right now? What is the problem? What is the question that we're wrestling with right now? Or maybe for the organizers as well.
I mean, when I read that, I wasn't quite sure what to make of it, so I just did something rather random. So, I was actually trying to look back over, I suppose, yeah. So I sort of grew up reading Provine's PhD thesis, which described this, synthesis of Mendelism with Darwinism. And I think many of us are familiar with that period up to the modern synthesis. And the past 60 years, if you look back at it, it's kind of quite peculiar because you have these parallel streams of, molecular biology being very impressive, amazing accumulation of really detailed mechanistic information. And also, I think, we should be proud of the conceptual advances that have been made in evolutionary biology. And I find it very hard to have any sense of where we'll go in the next 30 years, if that's. A lot of it depends on whether we can keep our memory of the past history and the broader questions rather than fragmenting into tinier and tinier little niches.
Can I ask a follow up?
Oh no, it's okay. I was just gonna say that my understanding was that that this was not going to be like a to and fro, but like a looking back and looking forward thing. So,
but if I can ask a question that maybe generates a bit of debate, what I've got from your two very interesting talks, it's a very different approach to what technology has done for us and what it is how it has really helped us answer questions. What do you think? Do you think that really technology is what has changed how we are answering questions or not? I mean, I know what you've presented in your talk, but can we think a little bit about how important it really? So what I think is that, of course, all the ideas are there and the way to test them are independent of the data. But the truth is that data is allowing us to answer some of these questions. So how important is technology and how important is going to be in the future versus how we answer it?
So I suppose I remember my naive enthusiasm in 1983 reading Marty Kreitman's work on balancing selection in ADH and Drosophila, really right at the beginning. It was the first study of sequence variation, when Sanger sequencing came out. And, there was a paper with a spike. And there was a spike, and you could see the balancing selection. And they were like, well, we're done. We can answer all this stuff. We just scan the genome, we find all the loci under balancing selection. That's it. And of course, it's turned out to be more complicated. Even that initial example of ADH, it turned out not to be at all Kreitman's initial explanation. And we still don't know what explains the molecular variation at ADH. But anyway, that's another story. So there was this sort of naivety towards just having data would answer it. And yet, of course, it's right that, we must be in a better position. I think we're in the very early stages of understanding how to use the data. And I think it will take much more hard work than people are maybe imagining, where you think, oh, you have a fancy software which will tell you the answer. And you can still publish papers with a fancy software that, will claim to find candidates and so on. But I think we have to move beyond that.
yeah, I think that a lot of the technology-driven science is dare I say it, quite boring. It's for me, And that there's no substitute for having, for having careful hypothesis making and testing predictions and having that conceptual overview. And there's too much. There's too much of biology being driven by technology, in my view. I think there's like not enough of that sort of conceptual grounding that I think evolutionary biology can provide sometimes.
I really liked the points you were making, Ashley, about the importance of imagination. And I think the way that technology helps us to imagine the world of other organisms, like particularly Toby's work, imagining trading networks in root systems and fungal systems, and the importance of drawing things and looking at them closely. And if that's what biology makes us do, and the excitement of being in rooms with biologists who'll talk about fungal fitness at the same time they talk about amphibian fitness. And I think the real dangers of taxon-specific science are really exemplified by where the huge bounds have been in evolutionary biology, which have been thinking across taxonomic groups and making these big comparisons. So I really the importance of imagination, and people like Haldane, for me, really embody that idea of looking at the world in the power of analogy, really. I really like that. Anyway, yes, more questions. Do you want a mic?
Microphone?
Seeing patterns in huge amounts of data, is that going to lead to any evolutionary breakthrough, or do we need creativity to come up with hypotheses and ask the right questions? So can AI find patterns in the data that will answer any of these questions or not?
Yeah, of course it can. But again, I think there's no substitute for having doing the work to think about what the question is and really, I know that I go to so many talks and just thinking well, what are you actually trying to? What's the question? What are we going to know after you've done this? What we didn't know before.
So the problem, as usual, is to know what question to ask AI in the first place.
Yes, I would say.
But I think there's a further problem, is that AI has to be trained on something that we think is grounded in truth. And we don't have any systems really where we know the truth. And, you see applications at the moment where people have the idea, which sort of makes sense unless you think about it for too long, of training the AI against simulations, and then you'll be able to fit the simulation model and it will find that very efficiently. But we don't actually know what model is appropriate. So I think there's a danger of being dazzled by the technology. Yeah.
Already have the microphone here. Thank you for you both for your talk. Since it's a debate, I'm going to try and be deliberately provocative. Nick, you were describing the modern synthesis, and you've just mentioned Provin. And it's funny because I was literally reading a paper before coming here, and I've been studying the history. Provin, Adams, Forsythe, they interviewed many of the modern synthesis people, Dobzhansky, etc. And they came to the realization towards the end of their career that the modern synthesis was not something that was novel. In fact, they were just getting rid of a bunch of different mechanisms that Darwin had proposed. And there was this apparent, I'm going to make it short, but they make the argument that microevolution, as in population genetics, leading to macroevolution was something that was assumed but not proved, because they wanted to shut down the creationists who were making claims, let's say, of the Cambrian explosion, etc. So my question to you is, do you think that microevolution, as in population genetics, changes in allele frequencies, can explain things like the Cambrian explosion, the appearance of whales from Artiodactylus in 7 million years, or it's time for us to put this old view of evolution to rest? And if it's yes, why are we still teaching this to students instead of, let's say, the extended synthesis and other views that put, let's say, the phenotype on top, for example?
I'm not quite sure what the question is, but the analogy would be with, whether, fundamental physics can explain chemistry and material science and so on. And of course, there are phenomena which, particle physics or Maxwell's equations by themselves are not very good at explaining. So in that sense, of course, no one's doubting that evolution is the change of allele frequency. That's, not up for argument. But whether knowing the principles of population genetics gets you very far in explaining the Cambrian explosion, well, probably not. It's a different phenomenon. And you have to know the constraints that are given by the underlying mechanisms, microevolutionary mechanisms, but then there are other phenomena that emerge, which will have different explanations, and maybe are not even ones where it's useful to think in terms of microevolutionary mechanism. So it's not that, yeah, it's interesting that if you look at the different fields, the fields I would have listed if the font hadn't shrunk, then actually a lot of the arguments in, say, the phylogenetic and macroevolutionary level are, to a large extent, independent of the microevolutionary processes. We have to have speciation rates and try and look in detail at what's going on at that speciation event. But over a large time scale, a branching tree works pretty well. So we have to use methods, concepts that are appropriate for the level and understand the connections between the levels, but also realize the levels are to some degree independent.
Yeah, more questions? It's the question down the front there.
Oh, thank you. Hi, I just wanted maybe to bring a positive note in terms of the importance of evolutionary biology in other disciplines. Just thinking of examples that I think really portray that and really recent and important advances, maybe on an applied perspective, but cancer evolution, evolution of antibiotic resistant, pathogen evolution with COVID. I think this is like a good several good examples how even society in general came to appreciate the value of evolutionary biology, and it has been transformative in these fields how we understand these phenomena. So I'm just curious to see any comments.
Of course, agree about the wonderful importance of insights from evolutionary biology in these fields. I'm not sure how widely appreciated in society it is. That's another question.
I'm so happy you said that because I think it's a really important point to make, and we were all really loving the fact that people in the street knew what R naught was for a few weeks, weren't we? I think maybe I would disagree a little bit. I think that probably the public did appreciate, they were able to see like in real time these like selective like sweeps of different COVID strains sweeping around the earth, and I think evolution was like really, sort of like smack bang in the middle of public debate for years. So I think that's it's a really good point. I mean, it's not a nice way to be brought to the public consciousness, but I think it has been a big impact, like you say, yeah.
It also kind of illustrated how fundamentally hard it is to make predictions in evolution. I remember all the physicists lining up to say, "Oh, this is all easy. We can model this." Why are you finding this so? Epidemiology just doesn't seem that difficult. And it brings me back to the question that I think comes from maybe both of your talks, which is that environmental variance is such a key critical. The pattern of environmental variance affects evolutionary responses, biotic interactions, abiotic interactions. And given we're entering a phase, arguably in the Earth's history, of very rapidly increasing environmental variance, big change in the environment, much more unpredictability, potentially, much more variance. Do you have a feeling about how you think that's going to take? Is that going to make evolution easier or harder? It's a very dichotomous question, but the way the environmental variance affects the ability of populations to evolve. No, okay.
Well, I mean, it's obvious that evolution, natural selection, wonderful as it is, isn't a cure-all. So clearly the environment can change fast enough that things just go extinct, and that's that. And of course, that's happened in the past. There not in my lifetime, but there have been big mass extinctions when diversity radically decreased due to catastrophes of the same order of magnitude that we're seeing now with temperature rises and so on. So, yeah.
This way that environmental variance affects heritability or genetic variance, the sort of the noise, if it's environmental noise, if it's kind of like predictable variation.
Yeah, but that seems a sort of second order concern. If the sea level rises, if temperature rises too much, then you're kind of dead at some point. And yes, your genetic variance might increase, and you might, be adapting a bit faster. But there are limits to what natural selection can do.
Absolutely, yes.
So maybe this is a bit philosophical, but with the fragmentation in the field, with all these subdisciplines and the overabundance of data and the need to look across broader scales of patterns, is a theory of everything in evolutionary biology possible? And if so, then what would that entail in terms of sort of bridging all these disparate questions together and sort of framing it in terms of a question with boundary conditions, whereas if you fall in this regime, these rules apply. If you fall in that regime, those kind of rules apply.
I mean, what do you think a theory of everything would look like? I mean, in some sense, Darwin had a theory of everything, a qualitative verbal argument that brought together lots of different phenomena. But I have the idea you want it more quantitative than that, or more detailed?
Extended evolutionary synthesis sort of thing.
But then what in, yes, I don't know. I'll give it
I mean, I'm maybe I'm not understanding your question very well, but I would say that like one of the strengths of our field is that we are so conceptually united and organized, like with this theory of evolution, right? So we could go into space and and construct sensible hypotheses about something we'd never seen before based on what we understand about evolutionary processes. So I think we do have a theory for everything.
The problem is it's contingent on environments, on the way organisms are constructed. So it can be manifested in very different ways. And yet, when you look at how we analyze very different situations like cooperation in bacteria, cooperation in meerkats, there's the same fundamental principles. And that, to me, is enough of a theory of everything. I wouldn't want it to all look kind of the same. That would be boring, like chemistry or something.
We've got a question down here. Someone's been waiting.
Hi. Kamal Ramsey, King's College. My question is that the study of evolution necessarily looks backwards in time. Is it possible to turn the lens around and look at the future of evolutionary processes?
Yeah, we can make we do that all the time in experimental evolution studies, where we're making testable predictions about how traits will evolve over time, and then we can test those in selection experiments. I don't know if that's what you mean, but I think that's one of the strengths of the theory, is that we can make these testable predictions about what we think are going to happen in the future. Yeah.
But it is very short term. We're talking about, tens, hundreds of generations in very well-controlled systems. Maybe the best real-world example of prediction would be in flu, where, people are reasonably successfully trying to predict what the flu strain will be in a year's time so we can design a vaccine. But it's very short term there. You could say that. I don't know. I mean, it's a hypothesis. Yeah.
Yep. So this goes to Ashley. So it's quite an interesting leap to go from the behavior of meerkats to bacteria, and I was wondering if you might speculate or comment on the things that would be lost or not by dividing behavior between those things that have neurons and don't have neurons. So is there an advantage to categorizing them differently or collectively calling sort of everything behavior?
I think that's where Tinbergen very much comes to the rescue, right? So the absence or presence of neurons is a sort of is a proximate, that's sort of like affecting our ability to make proximate causation arguments. But I think that but in terms of like whether, as Nick said, we can make exactly the same predictions. We have kin selection theory, we apply them to meerkats, we apply them to bacteria, whether or not they have neurons or not. So I think it's whether the exact way that they perform the behavior, whether it's neurologically determined, is much more of a proximate consideration, is what I would say. Is that, what do you think? I like a skeptical nod. I'd be interested to know what you think.
I don't know. I suppose you could reduce everything to sort of physical properties. But I guess I would tend to think that there are important contrasts to behaviors that emerge from brains or nervous systems than just pure sort of physical reactions.
Absolutely, but those again are like I'm not by saying they're proximate, they aren't, and that's not to denigrate them as not interesting or important. Far from it, but it's just I think that the Tinbergen system of like thinking about these complementary questions is a really useful one when we're thinking about those kind of considerations.
Thank you.
Time for a couple more questions.
Thank you. Very, very interesting talks. I wanted to ask you, to each of you separately, what do you think is the problem in evolutionary, like in our understanding of evolutionary biology nowadays, and how you will fix that problem?
What is the problem with evolutionary biology and how to fix it? Wow.
Yeah, that's yes.
Yeah.
Yes.
Yeah.
Other than making it what's the future, because that's very hard to say. But what would you really like to know about in 20 years' time?
Have you got a ready answer?
Well, not a very good answer. Well, I mean, so the question I was sort of circling around was just the question that was in my mind when I started out reading Launton's book, right? Which is just how much of this stuff we see, the variation we see, is shaped by selection. And I mean, related to that is really how to reconcile this very polygenic view of things where, there's very diffuse sort of function, very weakly perturbed by selection underneath, and yet on the surface it looks very deterministic, very much optimized by selection. How do we really reconcile those different viewpoints? It's not really a very clear question, and maybe the point is to try and sharpen that up and make it more precise and testable. But
I think that I've thought about it a wee bit, and I think it stumped me originally because I was casting around and trying to think of this big thing that I wanted to know, and I never have a good answer to that question, and I think it's because how I guess I proceed is just thinking about what I'm curious about today. So I'm not really thinking what is the big question. I'm more sort of guided by my own curiosity. And I could tell you what right now what that is, but I don't think I would ever have an answer to your question. Is the short answer, because it's not really how I think about things. Right now I'm really interested in Evo Devo and trying to think about the variation between there are lots of unexplained variation between the ways that cells compete with one another to be in the germline in multicellular organisms. I'm just, that's kind of what I'm interested into right now, but that's not necessarily what I think everybody should be doing.
Okay, there's two more questions.
Sorry, I want to follow up on that one very quickly. And what is the question that you think we're going to have an answer for in the next 30 years?
Yeah.
I think the thing that I've just the understanding the variation in developmental processes and the evolutionary basis of that. I think definitely we can work that out in the next 30 years.
You have to work on an evolutionary timescale. 30 years, not long enough. I mean, well, I go back to my anecdote of being super enthusiastic in 1983 that we'd almost solved the whole problem of the extent and the nature of selection, and here we are asking the same question. So I wouldn't want to be making any overconfident predictions.
Let's go one question then.
Yeah, I really liked the point that you said regarding the interdisciplinary missionaries for evolutionary biology. But I was thinking about that, and I was wondering whether, in either of your opinions, if if that's something that you want to try and do, whether it's more important to you yourself have more of an interdisciplinary understanding, or more important to just be more of an expert in the field, or alternatively, if it's just more important to be particularly outgoing, or yeah.
Yeah, I think it's really important to I mean, I in the last sort of 30 years or so that I've been doing interdisciplinary research, I think it's really important to choose the right collaborators. You know, just you have to get on with them, and I'm really of the opinion now that, you know, no science is worth it. No scientific discovery is worth the hell of having a bad collaborator.
Absolutely true.
So I think it's like, you know, it's like science is this, like, you know, one of the joys of being a scientist is it's just this really incredible, like, social enterprise, right, where we work together. When we were you know, when we're talking about this sort of crossover with these technology-driven, amazing science, you know, that's been done by molecular and cell biologists and so on, there's no way that we can learn that, you know. There's just none of us have time to just learn all this. We're completely reliant on collaborators. But like, you know, as Nick said, it's sort of like there's an asymmetry there, and in terms of like how the expertise is perceived and where the potential contribution is perceived. So yeah, I would just say, like, choose wisely.
Yeah, I mean, I completely agree that it hinges on having collaborators on both sides who are kind of open-minded. And, you know, I had a very negative experience with interdisciplinarity when I had this sort of strange idea of talking to computer scientists and talking about evolutionary computation, which is a whole field. It's, you know, there are 2 or 3 conferences a year, quite well attended, you know, a few hundred people studying how to, you know, give up on designing programs, but actually get them to evolve. You start out with a program that kind of works, you make random changes, and it's a very interesting field because they've discovered more or less independently that recombination is good. You must have too much mutation, but they don't have any quantitative theory, and they have tiny little theory groupings. And it turned out to be really, really hard to communicate because they have such a different style of research, a sort of a bit of a maths envy, wanting to prove things for the worst case, a certain style in computer science that was very different from the style we had. So that's, you know, an anecdote to say, yes, you need to find the right collaborators. But also to say that I'm not sure even that the missionary work out to the molecular biologists is the most important. I feel we also sort of have a responsibility to communicate really the nature and the wonder of evolutionary biology and the perspective it gives you on really how fragile life is and how, you know, extraordinarily brief the time span is that humans have been around and seeming to reach some kind of catastrophic singularity. And I think people are just not thinking about that. It's much less in the sort of general public consciousness than it should be.
Great. I'll take 2 more questions, then we're going to...
Yeah, that's it.
Yeah, thank you both for the lovely talks. You've both alluded to, you know, there's these satellite disciplines that think they're doing evolutionary biology a little bit. You've highlighted challenges in getting funding, other from the Royal Society. What do you think we as individuals can do now to, you know, ensure that big picture question evolutionary biology, you know, has a good future in this country?
I can say I'm not in this country, but
You guys are really putting us. It's a really good question, and I don't know, like, what, maybe you've got experience of this, but somebody once told me that, and this makes me laugh thinking about it. Somebody once told me that, like, if they're on an NERC panel, and whenever there was like an evolutionary grant came in front of them, there was like all the evolutionary biologists in the panel would just like absolutely hammer it and be like, you know, they'd be like, they're not doing this, they're not, yeah, there's control, but whereas other fields were much more positive about you know, anything to do with Antarctica would just be like, this is the most amazing thing. So I think we've got to maybe try and be a little bit more mutually supportive of one another.
Yes, I mean it's sometimes hard to bring oneself to be when you can see something doesn't work. But yeah. I mean, I suppose I have to put a plug in for ELC, which is one of the organisations that's been really good actually in funding basic research on big questions. And I think it's been a bit of a miracle that that got well funded, and it seems to have been a success. So, and it's very different from the landscape, I think, in the UK, where things are very, you know, focused on particular challenges which change, you know, with the government or the minister or whatever, making things very unpredictable and also forcing you to jump through hoops in order to do what you actually want to do.
Thank you. Actually, it was more or less on the same line. So most of the questions that have drawn many of us to this field of evolutionary biology is the sort of questions, for example, of the pervasiveness of natural selection, or how to reconcile this type of theories, for example, with the neutral type of evolutionary theory. Yet, as you say, Nick, this is the type of questions that have brought you like more than 30 years in your life, and yet you're still asking the same type of questions. As for us, as early research careers, we are facing an environment, for example, of funding of the whole research environment that is just like very short-sighted, in the sense that we just like find, for example, grants of 2, 3, maybe 5 years. And what are your thoughts on that? Do you think that this research environment is stimulating or is encouraging for us to pursue these sort of questions?
So there's different threads to that. One of the threads is that, you know, some studies need to be long-term, as it turned out our very slow Snapdragon study, you know, it's gone on 15 years, and that worked because we had we're lucky in ISD Austria to have some core funding that allows you to keep, you know, doing things. It's not that expensive, but still you need to have funding continuously. And there are lots of long-term projects which are really valuable and which really struggle, you know, to get continuous funding. I mean, how you cure the short-term visions of, you know, the higher administrations in science, I don't know. It's partly our fault for over-promising, for saying, oh, we can cure this and fix that, and so on, and get carried away in what we promise. And whether we can ever get back to a more sober and rational presentation of what really is the value of science and how it can work, and work, you know, slowly but not miraculously.
Yeah, I don't really have anything to add to that. Just as it's so important that policymakers understand the nature of science, you know, which they don't in the main. And the Royal Society, I know, like sort of has various programs to try and promote more understanding. I don't know if anybody's ever been on the MP scientist pairing scheme. Anybody done that? So I think it is like it's a really important problem, and I think that bodies like the Royal Society, I think, could be doing a little bit more of it, actually, to be honest. Yeah.
Maybe on that note. Yeah. Yes, exactly. So maybe on that note, we'll finish the debate. Thank you so much again for coming, and all of you. Remember to sign up to our mailing list if you are not registered with us. You just get a few emails of very interesting stuff, so you don't get a lot of emails. We have a hopefully wonderful reception that is longer than usual because we're celebrating the 30 years. So enjoy and see you soon, I hope.